class. This week we're going to be, for our last lab of this semester, we're going to be doing something a little bit different. We're going to be doing a biotechnology experiment. Some of you may not think about biotechnology as being chemistry, but it is a type of science that actually works at the interface of chemistry and biosciences. And a number of the things that we have looked at this semester, catalysis, rates of reactions, those sorts of things need to come into play when we look at some of the biotechnology techniques that are used. The specific example we're going to do this week is a DNA fingerprinting simulation. So we're going to be working with DNA, and we're going to be using some enzyme catalysts that are called restriction endonucleases to cut that DNA in specific sites. Now, we know that human DNA is about 90% identical from individual to individual, but there are certain places that are different, and that's what we're going to be using in this particular type of example. So you're going to be carrying out these incubations to cut some sample DNA in specific regions using these restriction endonucleases. And then we're going to separate the DNA fragments that we get by agarose gel electrophoresis. And then after some staining and destaining, you'll be analyzing the patterns that you see from that. Now, this particular video is just going to look at some of the techniques that we'll be using this week, since they're a little different than some of the previous labs. But the background information on DNA fingerprinting, and a little bit more about electrophoresis are found on some Panopto files that are also listed for this lab. Okay, so let's get started. First of all, to talk about safety, just as any chemistry lab, we're going to wear our goggles this week. We're working with small volumes of materials, and most of them are water soluble, but we are able to, we still need to treat them as if they are hazardous chemical materials. This is a simulation, so we're not going to be working with actual human DNA, so we don't need to treat it as a biohazard. But do keep in mind that if this was being done with actual human materials, then we would have to be collecting all the materials in biohazard bags and autoclaving it before disposal. There are a number of reagents and equipment that I'd like to talk about first, to make sure that you have them at your bench before you start working. And to know that this week we're going to be working with 4 people to a group. So each of your benches is going to serve as a group, and they will be running an electrophoresis gel. So let's start by looking at some of the materials that we're going to have in lab this week. Well, some of the first measuring devices that we'll use this week that you haven't used in the Gen Chem lab before are these micropipetters. And that's because we need to measure microliter volumes of samples this week. There are 2 different types of micropipetters that you may see in your lab. One of them is made by a company called Edvitech, and the other one is made by Rainin. But they both work pretty much the same. The one thing that you will need to remember is that if you have these blue Rainin pipettes, that you're going to use white pipette tips that will be available to you. And if you're going to use the Edvitech pipetters, we have the yellow pipettes tips that will be used for those. So I'll just demonstrate some things with one of these types of pipetters. So let's look at the Rainin pipetters. Now, as we look at this Rainin pipetter, you're going to see a number of things on here. There is a plunger. We've got the shaft of the pipetter. We'll note the silver that's on here. There's a little window that indicates our volume that we're going to be pipetting, and that is in microliters. The types of pipetter that you'll have in lab is a what's called an L200, and so it will measure the types of volumes that we need to use today. So let's say that we want to measure a 20 microliter volume with this pipetter. First of all, we've got this little knob up here, and we'll make sure that that's loose so that we can turn the barrel to change our volume on here. And so we're going to change this to 20. And as I said, this will do a maximum of 200. It actually will measure volumes between 20 and 200 quite accurately. So we can look at this and we can see that that is it says 200 there. A little difficult to see, but, and then we'll turn the knob on top here so that kind of holds the plunger in place. Now we have the pipette tips, and we'll have some of them in boxes like this. What you'll want to do is gently put the pipetter into the tip in the box, but then you do want to make sure that that is firmly on the end by pushing that with your finger and giving it just a very slight turn. We don't want to break the barrel of the pipetter. Now, anytime we do micropipetting, this seems like it'd be easy to do, but to do this accurately, we need to follow some guidelines. There is, if you play with the plunger, you'll note there's a first stop and a second stop. What you want to do when you take up volume is go to the first stop in the pipetter, put the tip just beneath the surface of the liquid that you're pipetting, and let that up slowly. And you should see liquid in the pipette tip. Now to dispense, however, we're going to put our pipette tip in our microfuge tube, and we will not only go to the first stop, but the second stop. And that should accurately measure our volumes into our microfuge tubes. This shows another thing that we're going to use this week. Our chemistry is going to be done in these microfuge tubes that we won't have to clean test tubes this week. These are disposable. Do be careful with the pipetters that you don't lay them down with liquid in them. We never turn them upside down with liquid in them, because if we get liquid in the barrel of the pipette, that has to be fully cleaned. If that has to be done, if it has to be sent back to the company to be done, that can be a $100 job. Now, these pipetters are well over $300 a piece, and so we're trusting that you're going to handle them carefully and use them responsibly in lab this week. The way you get your reagents this week might be a little bit different too. You'll have a lot of things in small tubes. There are some DNA samples that are said to be the crime scene DNA, and those will be available to you either in a small tube like this, or they may be given to you in a card like this, and those are quite nice because then the card just has the sample that you need and you'll pipette from there. You'll also have samples at your desk of the restriction enzymes, enzymes 1 and 2, and they will be labeled. You'll also have the suspect DNA, DNA 1 and DNA 2 that you'll be using. You will need to have 4 reaction vessels, 4 different microfuge tubes. And as I suggested, it would be good to label them, obviously, so that you can keep track of them. And a good way to label them are suspect 1, reaction 1, that's why I suggested S1, NR1, suspect 2, reaction 1, S2, R1, and the same thing for the other sample. But you'll have 4 of them. Your suspect DNA will be treated with 2 different restriction enzymes, but each of which catalyzes the hydrolysis of DNA at a different site. Now, very small volumes that we're using, and in these small tubes. And if we were pipetting into these tubes, so we're going to have a number of things. So as we said before, we're going to push the plunger down, put that under the liquid and just the surface of the liquid, let the plunger up, go into our next tube and plunge down to the second stop. That might be where we add our first DNA. And then we'll add our reaction buffer, whatever volume that is supposed to be, and our enzyme. And remember, I didn't at this point because I'm saving on tips and this is for demonstration, but anytime you're putting these tips into a new reagent, you should change the tips that you're using. We will have waste containers for the tips. This white on this particular pipetter, this white button, that ejects the tips. On the Edvitech pipetters, it is the blue button that's right here. Well, now we have this sample, but we've just put in what seem to be just drops of material. We need to make sure that everything is mixed so that it will react. So we can close that, and we've got two samples here. I've done another one earlier. So we have these two microfuge tubes that have sample in them, but we've put such small volumes in, the material may not have all gone to the bottom of the tubes. So one of the things we can do is tap that on the bottom of on the bench, or we can use one of these microfuge centrifuges that are here. Again, as with all centrifuges, we're going to have to put those tubes across from each other to balance them. This centrifuge at high speed will go about 14,000 rotations per minute, and so we really need to make sure that those are balanced. We have a lid that goes on that. Close the lid, and just a momentary centrifugation will bring all of that solution down to the bottom of the tube. And then we're ready to do our incubations. Our incubations are going to be done in a water bath that's kept at 37 degrees. And what we'll have are these floating devices that you can put your tubes in, and then just float that in the water bath. Just float that in the water bath and leave it for the period of time that is required. While your samples are incubating, you're going to have a chance to practice loading a gel, because sometimes pipetting with these pipetters in these agarose gels is a little bit tricky. So you'll see at your station that you'll have a practice gel loading solution, and that's what you'll use for that. The gel that's provided to you may look like this, or it may already be out of the forms. For time's sakes, we decided to make the gels for you. But what happened was that we made a 0.8% agarose slurry in our electrophoresis buffer and poured it in these forms, these plastic forms that have these rubber dams on either side of them. And then while it was still liquid, we put this comb in. This comb was going to make it so we have 8 nice wells for doing our sample loading. And so we can see that a little bit closer if you'd like. If you were pulling these combs out, you would pull them out just straight up and down, because we don't want to break the gel. And we should have lots of nice little 8 little wells across here. The other tricky part is to get these dams off without losing our gel, and so we want to take those off carefully. And so we have an agarose gel that looks like this. Now these all go into our electrophoresis unit, and that's what we have here. This blue electrophoresis unit, and we'll have one for each classroom. And you'll be able to tell that you have your gels in correctly because they have little notches where they fit. Some electrophoresis buffer will have been made for you. And anytime we're going to add samples to our unit, we're going to make sure that we have all of the gels submerged in that buffer. They all have to be wet. They all have to be just under the buffer just has to go over the surface of the gels. Now, while you're waiting for your samples to incubate, you can use this practice loading solution to do some pipetting into these wells. That's a good idea so that you have practice and make sure you haven't punctured the wells and things like that. And then you'll just take that gel out and we'll throw those away and we'll put in the good gel for our samples. Unless it still looks pretty good, then we may just rinse it out and use that one for your actual electrophoresis. But we'll do some sample loading while you are waiting for your incubation. Well, we want to stop our samples from reacting too far at the end of that 30 to 60 minutes. And so what we'll do is we will add this gel loading solution. This gel loading solution has reagent in it that will denature the enzyme, so it will stop that enzyme catalyzed hydrolysis of the DNA. And it has a dye in it that will migrate faster than anything else in our samples on our electrophoresis gels. And so we'll be able to monitor when our electrophoresis is completed when the dye front just about reaches the end of the electrophoresis gel. So when we have our reactions, we're going to add the required amount of our gel loading solution to that sample to stop the reaction. Make sure you cap everything. We have very small amounts of reagents, but it will be oodles for all of our classes as long as we're careful with everything. Tap that down, or alternatively use the ultracentrifuge to microcentrifuge to mix your samples. And now we're ready to apply these to our wells. The volume that we use that's suggested is 40 microliters. And so you'll want to set your plunger for 40 microliters, so it says 40. However, it's not necessary that that's exactly 40, but that's about the amount that will work best for this. And we'll put our plunger down to the first stop, into our sample, just under the surface, let it up, and we're going to apply that to our wells. Now, these are hard to see. Sometimes students have trouble seeing these, and what I would suggest is that you check to see whether you like this under a white sheet of paper or on the black surface. And the important thing for this when you're pipetting this is don't let that plunger up until you have removed that sample from the well. Otherwise, you're going to blow bubbles in there. So you want your sample to go into the well, but not puncture the gel. Or have it float away. Now, in the interest of time, it could be that we have a lot of samples to apply and not enough pipetters. In that case, we have some single volume pipetters here that are available from Edvitech. They are these small purple ones like this. They're only 30 microliters, but that is still good enough volume. The yellow tips fit on them, as you see here. And for these, we just go down all the way, suck up our sample, and we can apply to our wells. There is a little glycerol in this sample so that the sample tends to sink in our solution. Be sure if you have any questions about applying these samples or doing any of this work here, that be sure to ask questions. That's perfectly fine and good. So if we look at this a little bit, I can get a little bit closer here. We can see the wells of that electrophoresis gel have been filled with our reaction mixture or our sample if we were just practicing at that point. Now that looks really, really good. Now once everyone has their gels loaded, we want to put the cover on the electrophoresis unit. Make sure that we have the black terminal on the black end, the red terminal on the red end. The buffer that we're using is such that all of the DNA fragments are going to be negatively charged. So we want them to set them in this unit so that they are migrating toward the positive pole. And then we have a power unit here, and again, we're going to want to make sure this is hooked up so we have black to black and red to red. And then turn on our power source here. Here's our electrophoresis unit. And we wanted to read voltage. So you can see here that there's a little yellow dot here by a V, indicating that it's reading voltage. We also see a yellow dot here, and we're going to want to set this. And what I suggest is that we set this at 150 volts. So we're going to use the toggle arrows here to do that. And that's set on 150, but it hasn't started yet. When we want it to go, we're going to hit this little running man here, or the on button. And we should see that that should go up to 150. The other thing that you should notice if you have everything hooked up correctly is that when you look at your electrophoresis unit, you should see bubbles in the system. There will be bubbles at each of the electrodes. This is what's happening is we're putting electricity through the buffer, which is mostly water, and so we are getting electrolysis of that water. Those bands should then migrate from their wells and move down toward the opposite end of the gel. If you see that they are moving in the opposite direction, you have the system set up incorrectly. And stop it before you lose your sample. In the olden days, they didn't use to color code these wires, and there were times that we set up electrophoresis units, turned our backs, and our sample had migrated out the top and into the buffer. We don't want that to happen. After the bands have just about reached the end of the gel, don't let them run off, but after they've just about reached the end of the gel, then you will want to turn off the power on the power supply, unplug the machine, and then unplug the top of the electrophoresis unit from the holder. Then you're ready to take the gel out and stain it. There are some kinds of stains where we're not going to want to touch this with our hands. However, this one should be just fine. So we'll be able to take it out and place it in our Tupperware container or in a large weigh boat, either way. And so we'll have that down there. Now we're going to do staining very easily. Sometimes you can actually pour stain into your container. I'll show you what this looks like in here right now. Just got this in the Tupperware container. Sometimes you can, most often in the research lab or whatever, we're going to use just a liquid material or liquid stain in order to do this. But for our work here today, we're actually just going to use a very simple system that involves the use of what's called a card. Let's move this buffer out of the way here. We'll move our Tupperware container a little bit closer. But what we'll do with this is that we will take a card, and this is called Instastain stain. And it is going to we'll just be able to put that directly on our gel. As long as it's wet, we're just going to be able to put that directly on there. And probably use just a test tube to roll that on there a little bit better. A small test tube might work. Alternatively, we could just use a glass rod or something like that. But we want to make sure we've got good contact of that stain with our gel. And then after about 10-15 minutes, we're going to be able to peel that away. And you'll see that the whole gel is blue. But what we'll do is we will remove any of the excess dye by rinsing that with some warm distilled water. And then what should just be remaining are our DNA bands. After that, we're going to put the gel on a light box and take a picture of it, and you'll be able to do the work with analyzing your fragments to see which of the suspect DNAs looks more like the DNA that was collected at the simulated crime scene, and you'll be able to measure the relative mobilities of each of the bands in order to determine the relative mobility. I don't have an actual gel to show you, but after you've stained your gel, you should see a pattern something like this. That in your well where you had your DNA standards, you're going to have a number of bands. We'll have the sizes of those up on the board when you do come to lab. And then for your various samples, your crime scene cut with enzyme 1, your crime scene cut with enzyme 2, your suspect 1, 2, suspect 2, 2. You'll have a number of bands that you'll be able to see. And we're going to determine we're going to analyze these both qualitatively by comparing our band patterns to the DNA that was collected at the simulated crime site, and we'll also analyze them for their size by comparing to these size standards that we have on the left. Our total maximum distance that could be migrated comes from our center of our well to our dye front, and as I said, we don't want to let that run off, so we want to make sure that we have that blue band along the bottom. And then that's our maximum mobility. When we measure from the well to our band of interest, let's say that we're measuring for this band right here, that's our migration. To determine the RF, we're going to divide the migrated distance divided by the max distance, and that's going to give us our RF, and it's always going to be less than 1. So you'll determine that for a number of these samples that are on here. As I said, it will be our maximum distance is to the dye front. We'll measure the distance from the well to the middle of our band of interest. That's our migrated distance. Our RF is going to equal our migrated distance for a specific band divided by the maximal distance that could have migrated. And RFs will always be less than 1. Then you'll be doing some work where you will be plotting the sizes of the DNA fragments for these size standards that we have here versus the relative mobilities for each of them, the RFs, and see which graph, whether it's number of base pairs versus RF or log of base pairs versus RF, gives you the best situation for being able to predict the sizes of some of these other bands. As usual, you'll do that on Excel. For the linear relationship, you'll determine the equation that describes that, and then you'll be able to measure the RFs for these other bands, plug those values into that equation, and determine the size of those fragments. What we have with electrophoresis, as you note from reading and listening to the introductory information on DNA electrophoresis, is that the smaller bands will migrate the furthest, the larger bands will migrate the shortest distance during the electrophoresis. So, at the end of this week, you will have carried out an enzyme-catalyzed cutting of DNA, made some comparisons of 2 different DNA samples to a simulated crime scene DNA. The fun part is to be able to look at those patterns and see who was the suspect, who actually seemed to commit the crime. You'll have run a DNA electrophoresis gel, an agarose gel, and looked at that technique. That's something that you'll be using multiple times in both your bioscience and chemistry classes that you take. And we'll do some analysis of those patterns by measuring the relative mobilities of those bands, and doing some graphing to see the relationship between the size of the DNA fragments and their relative mobilities on those DNA gels. So hopefully this was an enjoyable end of the semester lab, and thank you for a good semester.